Hello, friends on the internet. Well, today we're going to be studying plasmid mapping. Yay. Let's look at number one. Here's number one. Well, there's number two as well. Number one, Walker, read that for us.
PGen 101 is a 20 kV plasmid.
Oh, let's stop there. PGen 101. What does that mean? It's the plasmid. It's named Gen 101. Good. So, PGen 101. So this is a plasmid, okay? And it's a 20 kilobase pair plasmid. 20 kb kilobase pairs. What are base pairs? AT, C's, and G's. So there's 20,000 base pairs in it. That's how big this plasmid is, okay? Big. Lots of base pairs, okay? So it's, we'll say 20 kb. Okay, so that's what we know about it so far. Keep going, Walker.
When digested with eco-RI, one linear fragment results.
Okay, let's break that down. What does it mean when it says when it's digested?
Let's take it in.
Digested means it's not digestion like in your bodies. More specific. When it's cut with a restriction enzyme. Good. When it says digested, when we're talking about plasmid mapping, it means when it's cut with the restriction enzyme. Okay? So, when it's cut with EcoR1, that's a restriction enzyme from E. coli that scientists have taken and used. It cuts this plasmid how many times?
3.
Are y'all high? It says one linear fragment. So if it's like round and you cut it and it results in one fragment linear like a string, you would only cut it once, right? So it's cut somewhere on here, okay? Now, the whole plasmid mapping is you're mapping out where the plasmids get cut with the restriction enzymes, okay? So that's what we're trying to attempt to do. Okay. Read the next part for us. Walker, keep going. You're good.
Digestion with BAMH1.
All right, what's BAMH1? It's another restriction enzyme. Dang, y'all are good. Keep going.
Results in 3 fragments of the following sizes: 12 kilobase pairs, 2 kilobase pairs, and 6 kilobase pairs.
Okay, so how many times did it cut it?
3.
3. So at 3 different places with this enzyme. Because remember, restriction enzymes are like engineered by the bacteria to cut DNA at a specific point. So it just so happens that BAM high, which is I'm going to call it for short here, it's cutting it, it's recognizing that sequence that it cuts at 3 different places on this plasmid. Does that make sense? So it's cutting it in 3 different places. I'm going to go ahead and kind of split it up here with the BAM high. So cuts it at 3 places, and so I'm going to say the one here, we'll say BAM high. It doesn't really matter where. You can think of it like a clock or something, like you could turn it one way or the other. We're looking for how does it cut it, how does the restriction enzyme cut it relative to the other restriction enzymes, okay? So it cuts 1 12, so half of it's 10. So we're going to say this one's 12. And then it cuts to what are the other 2 sequences?
2 kilobase pairs and 6.
2 and 6. So we don't really know. 2 could be down here, 2 could be up here, 6 could be switched back and forth. It really doesn't matter since we don't know more than that. Okay? So 12, 2, and 6. Does that make sense? Okay. So we've just cut it up into 3 different segments. That's really all the information we have so far. Now, Katie, what's the last thing that tells us?
Combination digest with eco-RI and BAM high.
What does that mean, a combination digest? So it mixes the plasmid with the 2 restriction enzymes, and they're both cutting it now at the same time. Okay? So how many fragments do we end up with?
4.
What?
4.
4. And they're what sizes?
8 kilobase pairs, 4 kilobase pairs, 2 kilobase pairs, and 6.
Okay. So how many times did EcoR1 cut it? Well, we've already got listed out where all our BAM cuts are, right? Okay. So how many times did EcoR1 cut this? Once. So we've got to find the one spot on here where EcoR1 cuts it relative to where the BAM high cut it. Okay? So, Tay, where do you think EcoR1 cuts which one of these fragments? into?
Where?
Yeah, where?
On the opposite of bam high? Like, on the opposite side?
Well, I mean, there's three of them. What do you mean opposite?
Like, the reverse side. I mean, like the opposite of each one. I don't know,
No.
it's confusing.
Katie, what do you think?
Does
So-
it cut,
Yeah, go ahead, Katie.
does it cut somewhere where like 12 is?
Why do you think it cuts to 12?
Because you get one that has 6 instead of 12. Like when we.
Well, the BAM high had a... Okay, so when you list it out, when you cut it with BAM, you wound up with a 12, and this may be a better way to write it out, a 6 and a 2 fragment, right? Okay? When you cut it out with Eco R1, you wound up with a fragment that is 20, so it only cuts it at one spot. But when you cut it with both of them, you wind up with a, what, 8, a 4, a 6, and a 2. So where do you think the Eco cuts that?
Somewhere in the 12.
Somewhere in the 12. So it's cutting the 12 here into an 8 and a 4. So when you added that Eco, the 6 and the 2 is still there, but that 12 is no longer there. It's switched into 8 and a 4. So how are we going to cut that, Mason?
Somewhere like 1/3 of the way.
Somewhere on the 12, yeah. 1/3 is going to be 4, and 1/3 is going to be, I can do it up here, it will be 8. And it could be the 4, you could have the 4 down here and the 8 up here. We don't know. We don't have any other information to go off of. All we know, and all you need to know, is that it cuts that 12 into a 4 and an 8. Does that make sense? So you've mapped out your plasmid. You know it's cut it at this many places. And you know the Eco R1 split that 12 into an 8 and a 4. And that's all the information we have, and that's all we can do with that one. Does that make sense? Okay, so there's another one at the bottom. And let's see if you guys can work on that and do that for homework, and we'll post a solution later.